Good afternoon again to everybody. We can start with the second part of this first day of the workshop. And we start with the first speaker, is Professor Cattaneo, Elena Cattaneo. I was very impressive from the curriculum. It's very interesting. The Professor Cattaneo worked at the University of Milano, the Department of Pharmacological Science, and she's a director of the Laboratory of Stem Cell Biology and the Pharmacology of Neurodegenerative Disease. She has a very big group, about 25 people. She spent a longer period in the Department of Brain and Cognitive Science in USA, recently she is also co-founder of the Center of Stem Cell Research of the University of Milano. The presentation of the Professor Cattaneo has the title BDNF and cholesterol dysfunction in Huntington disease. So please go on.
Thanks a lot, and thanks for this very kind invitation. I'm going to talk to you about one of the two main lines of research of our lab, which is Huntington disease. So we're mainly interested at the mechanisms in Huntington disease and also at finding possible cures for this neurodegenerative disease. And if we go to the next slide, which is a movie, so basically this is to present to you, this disease. It was also known as Huntington chorea. Chorea from the Greek means movements. And indeed the patients move like, as you see here, basically for the entire day. They are unable to remain sit on a chair for more than 2 minutes. So it is classically a motor disease that appears around, so the mean age is 35 years of age. After a number of years, the disease becomes also neuropsychiatric. So there is the appearance of depression and also psychiatric dysfunctions. So the incidence, so this is considered one of the rare diseases, but indeed it is absolutely not rare. In Italy, there are 5,000 individuals that are affected. And since, as you will see in a second, that this is a genetic disease, you can imagine that, of course, in addition to these affected patients, there are a number of relatives that are at risk. So overall, we count, at least in our own country, an approximate number of 30,000 people that are in some way affected by the disease, and you can make your own counts for Europe and USA. As you have seen, the clinical manifestation include choreiform movements, and then, as I said, cognitive impairments and emotional disturbances. The neuropathology is quite specific, so it is a brain neurodegenerative disease. The main brain region affected is the striatum, so the striatal neurons are preferentially affected. Although the more we study the disease, the more we understand that indeed there is a generalized neuronal dysfunction and neuronal degeneration. Although, again, the striatal neurons, at least at the beginning of the disease, are preferentially affected. And if I go to the next one. As I said, this is a genetic disease, and normal healthy subjects have 2 copies of the normal gene, which is called Huntington. And this gene is located on chromosome 4. And in 1993, it was discovered that Huntington disease is due to a mutation in the 5' end of the gene, and the mutation is an expansion of CAG repeats above 36. So below 36 is a normal allele, above 36 is a mutant allele. The CAG is translated into glutamine in the protein. So the CAG gives rise to the Q, and Q means glutamine. Therefore, the protein, which is indeed called Huntington, the mutant Huntington protein, is characterized by a number of Q, therefore of glutamine, above 36, and the normal Huntington protein is characterized by a number of Q below 36. So everything starts because of this CAG expansion in the gene, and geneticists correctly, of course, say that this is a dominant disease because one copy of the mutant gene is sufficient to cause the disease. And they are absolutely right in saying that if you want to study Huntington disease, you have to understand what mutant Huntington does, because the disease starts because of that CAG expansion. And the CAG expansion, indeed, defines the onset of the disease. The longer is the CAG expansion, the earliest are the symptoms. So there are cases of patients with 90 or 100 CAG repeats, and these are juvenile HD cases, and sometimes this occurs also in kids that are 5 or 10 years old. Although geneticists are absolutely right that, of course, mutant Huntington is a species, the protein that has to be studied if you want to understand Huntington disease, we are biologists, and therefore we try to approach the disease in a different manner. And if you are a biologist, basically you want to know what the normal proteins do physiologically in our body or in our neurons. And of course we were very attracted by Huntington and by this paradigm, not only because of what it means to the patients, but also because if you think at this scientifically, it is absolutely intriguing that here you have a case of a protein that is normally present in our cells, that when mutates causes neuronal degeneration, and remarkably, this protein and the affected gene is, of course, expressed in all cells of our body organism. Yet, only some cells are affected. So we were very attracted by this paradigm because we thought that through studies on Huntington disease, we could understand better, events like selective vulnerability, so how it is that mutant Huntington is expressed in every cell, even in fibroblasts, even in blood cells, but only neurons, and especially the striatal neurons, are preferentially affected. So again, this is just to give you some background. But our working hypothesis started from, I would say, some very basic biological questions. And the question here is, so what is the function of normal Huntington? And again, if you raise this question in a context of, in an audience where geneticists working on Huntington disease are present, they would say, who cares about normal Huntington? Because the disease is actually caused by the mutant protein. So at the beginning, it was actually difficult to start this program, but I can tell you that in the end, in several of our coalitions, many of these geneticists are actually involved, and are also studying with us the function of normal Huntington. So our idea was that normal Huntington that is expressed in all cells of the body organism must do something, let's say, general in all our cells, but maybe normal Huntington has developed some special function in brain neurons. So we want to know what are the functions that are not brain-specific, and what are the functions of normal Huntington that are brain-specific. And second, we want to understand whether or not there are functions for normal Huntington that, if lost due to the mutation, could contribute to aspects of the disease, like the selective vulnerability. So when we started, and actually I should say, our experiments and the experiments that you will see are really very simple, because all we do is basically to overexpress normal Huntington, to overexpress normal Huntington or to reduce normal Huntington levels in cells or mice to try to understand, what is the phenotype and what are the molecular pathways affected. And secondly, we are also interested in raising normal Huntington level. Of course, our idea is that normal Huntington is beneficial. So that was the working hypothesis. And if it is beneficial in some way, is there a way that normal Huntington overexpression could rescue from toxicity caused by the mutant protein. No? Because in the patients you have the normal copy and the mutant copy. How about if we are able in some way to raise the beneficial function of normal huntingtin? Can this compensate for the toxicity caused by the mutant protein? And of course, the other experiment is that if you reduce normal huntingtin level, can you worsen the disease phenotype? So this is our scheme. And I show you just already one result without showing you the data, but this was an old data. So we first found that normal huntingtin indeed is a very interesting protein because it is an anti-apoptotic protein, I would say neuroprotective, both in vitro, so we generated the first data in vitro, and also in vivo. And this was reproduced by several colleagues in the world and using different systems. So this was the first indication that normal huntingtin is an interesting protein, is beneficial. But then, I mean, our question was, is it possible that loss of this anti-apoptotic function of normal huntingtin in HD could actually be responsible for the selective vulnerability of those striatal neurons? And our guess was that that was rather impossible because if normal huntingtin, I'm not showing you the data, but if normal huntingtin blocks the apoptotic cascade we know, how this apoptotic cascade work, they are definitely not cell-specific, they are not brain-specific. And actually the idea that we have in the lab is that this anti-apoptotic function of huntingtin is something that occurs in all cells of the body organism, and this may be the ancestral function of huntingtin when it was born 250 million years ago. So, but then, that was very inspiring because we started thinking that maybe in addition to this anti-apoptotic function, huntingtin could also have some other brain-specific beneficial function. And this is when we started in 1999, when Chiara Zuccato joined the lab, and among the several possibilities, this was the very lucky one. We started thinking that we were actually intrigued by the fact, and actually we know that the striatal neurons that sit here and that die in Huntington disease depend for their survival on the delivery of a very important neurotrophin, which is called BDNF, brain-derived neurotrophic factor. And the BDNF is produced in cortex and is delivered to the striatal neurons through this cortico-striatal connection, anatomical connection. So the BDNF is made here, is transported and released at the level of the striatal neurons. The striatal neurons require it for their activity and survival, but they do not synthesize the BDNF themselves. They depend on the supply from cortex. And very interestingly, although normal huntingtin is expressed, I would say, at high level in the brain, it is actually very abundant in these cortical neurons that project to striatum. So the question is it possible that normal huntingtin here in these cortical neurons has something to do with the production or delivery of BDNF from cortex to striatum? Because if this is true, then we can imagine that when huntingtin is mutated due to the CAG expansion, then maybe this function, the production or delivery of BDNF, may be disrupted, and therefore less BDNF may be delivered to the striatal neurons. So this was our hypothesis. At that time, we were looking for additional neuronal specific, I would say striatal specific function of normal huntingtin. And we were not wrong, although, of course, it took a long time, to get all this data. But again, since we are cell biology, we started from scratch, from very simple experiments, and these are cell lines. These are parental cells that we generated, actually that I generated 25 years ago. These are immortalized cells from the rat striatum. And then what we did here, we simply overexpressed the normal huntingtin protein in these cells, or we overexpressed the mutant huntingtin protein. And then we produce a large number of clones. And then what we did here, we measured the level of BDNF. So how much BDNF are these cells producing after overexpression of wild type or mutant protein? And you see immediately, if you want to capture the message of this talk, you see immediately that overexpression of wild type huntingtin causes an increase in BDNF production, while overexpression of mutant huntingtin actually doesn't have this effect and actually is below the control. So the conclusion here was that normal huntingtin has something to do with the production of the BDNF. Okay, but this is in cells in vitro. And in this case, we were dealing with cells overexpressing normal or mutant huntingtin. So it is a rather artificial system. So we wanted to test another cell system. So the next cell system that we tested for the level of the BDNF are these cells. So these are again cell lines. 7, these are again parental cell lines that were produced from mice, just normal mice, that have 7 CAG in one allele and 7 CAG on the other allele. So mice have 7 and 7 CAG. And these are cell lines that were produced from knock-in mice made by Mercy MacDonald. So these are mice in which a 109 CAG repeat was inserted into the endogenous locus of the mouse. And in this case, the production of this mutant allele and of this normal allele is at physiological levels. Okay, so we are not overexpressing. And this is the homozygous knock-in, okay, 2 copies of mutant huntingtin. And here, as you can see, as you go from 2 copies of wild type to 1 of wild type to 0 of wild type, there is a progressive reduction of BDNF. So again, it seems that there is a link between huntingtin level and BDNF level. But again, geneticists will tell you this is not really the effect of the loss of normal huntingtin. It could actually be this reduction of BDNF. It could be due to, again, a toxic activity of the mutant protein, because here you have 2 copies of the mutant protein. So we had to do another experiment to convince ourselves and the colleagues. And the experiment here is done in embryonic stem cells. In this case, these are embryonic stem cells, just normal embryonic stem cells, that, from mouse in this case, that carry just the normal huntingtin gene. And these are embryonic stem cells that are heterozygous knockout. So there is 1 copy of normal huntingtin and the other is removed. And this is the homozygous knockout. So there is no huntingtin at all. And as you can see here, if you measure the BDNF, as you go from 2 copies of wild type to 1 to 0, there is a progressive reduction in BDNF. So there is a link between huntingtin, normal huntingtin level and BDNF level. But we wanted to do more, and then we subjected these same embryonic stem cells to a neuronal differentiation protocol. And as you can see here, and as it is known in the literature, when you differentiate these cells into neurons, so these are the parental cells, the level of BDNF goes up around day 14, this is known. When you do the same experiments with your heterozygous or homozygous knockout cells, you see that the cells are, again, less capable of making the BDNF. So the conclusion was that, at least in vitro, there is a link between huntingtin, normal huntingtin level and BDNF, and when there is the mutation, the level of BDNF is reduced. This is in vitro. How about in vivo? Because again, we are all always worried that this is an in vitro artifact. In vivo, fortunately, we had the possibility to use mouse models that were produced in this case by Michael Hayden in Vancouver. And as you can see here, there are just normal mice. And then Michael has produced mice that are overexpressing the wild-type huntingtin protein, so overexpressing. We are basically in the same condition as in our cell system in vitro, but in this case, of course, we're dealing with mice. Overexpressing wild-type huntingtin with 18 CAG or mutant huntingtin with 72 CAG. And what we did here, we measure, again, the BDNF, of course, now in cortex versus striatum. So if you increase huntingtin level in the mice, do you have more BDNF in the cortex of these mice? And if you overexpress mutant huntingtin in these mice, is there a reduction in the capability of the cortical neurons to produce the BDNF? And the answer is yes. I mean, if you focus just on this panel, cerebral cortex, these are the normal mice. These are the mice that overexpress wild-type huntingtin. And you see that these neurons, this cerebral cortex, produce more BDNF. And in the mice, in the cortices from mutant huntingtin mice, this effect is gone. And if you now go to striatum, you should expect that there is more BDNF received in striatum from cortex in the wild-type huntingtin overexpressing mice, and indeed this is the case, and there is much less in the mutant huntingtin mice. So this was very reassuring, but this was a first mouse model that we used, and we added more data. I'm not going to show this to you, but at some point we ask whether how exactly normal Huntington is regulating the production of the BDNF, and the message was that normal Huntington, I will show you some data but not all the data, that normal Huntington stimulates the production of the BDNF protein by increasing the transcription of the gene. So there is more, if you overexpress wild-type Huntington, there is more messenger RNA for the BDNF and more protein. If you overexpress mutant Huntington, the transcription is shut off, and therefore there is less BDNF produced. So, and I will come back to this, but I just want to show you that at some point, this was 2008, we wanted to see whether something like this was occurring in HD patients. So we wanted to test cerebral cortical cortices from cortex from, this is postmortem human cortices from HD patients. And of course, with the caveat that you can imagine when you deal with postmortem tissue, yet, I mean, we measure this is a control and this is the HD patient, this is a protein. You see that there is less BDNF protein in cortex of HD patients. And if you measure the messenger RNA, of course, this is also reduced. So, and the other thing that we did, ourselves and many other colleagues, so there have been really a lot of confirmatory studies. Of course, they became confirmatory, but afterward. But several groups have tested this idea of normal Huntington increasing BDNF and mutant Huntington affecting BDNF gene transcription and production. And I would say that this is probably the most consistent data in the HD field. So basically, every cell line, every mouse model that have been tested by every group in the world have confirmed that there is a link between Huntington and BDNF, and in the presence of the mutation, there is a reduction. So this was very important, of course, to progress with the studies. So not only in Huntington is anti-apoptotic in vitro, but Huntington has a very special function in the brain that has to do with the production of the BDNF. Normal but not mutant Huntington stimulates the production of BDNF by increasing its transcription. And we're shown, and this is published in vitro and in vivo, and I've shown you that human HD patients, have reduced BDNF in cortex. So the data seem robust, to the point that some colleagues started asking the question, okay, but what is the real impact of BDNF depletion or increase in HD? I mean, if this story is true, you would expect that if you remove the BDNF in HD mice, you should worsen the phenotype, you should see a worsening of the phenotype, and instead, if you increase the BDNF level in the mice, you should improve the phenotype. And also, the BDNF story survived to all these tests. And for example, one group has shown that if you cross mice that are BDNF heterozygous knockout with HD mice, so basically you are reducing the BDNF level in the HD mice, there is a worsening of the disease phenotype. In 2008, another group showed that if you cross BDNF overexpressors, so these are genetic evidence, if you cross BDNF overexpressors with HD mice, you improve the pathology. And very importantly, there is also these other studies, basically these groups have done a very interesting experiment. So they have removed the BDNF in cortices in the mice. So they've done a conditional BDNF knockout. Okay, so there is no mutant gene around, okay, just normal mice and removing the BDNF. And very importantly, they discovered that there is striatal neuropathology. So removing the BDNF from cortex is detrimental to the striatal neurons. And there is an HD-like behavioral phenotype. And if you compare the gene expression profile that you get from this BDNF knockout with the expression profile that you get from human HD cortices, okay? Basically, this gene expression profile is almost overlapping, okay? And it is the most overlapping. So they compared also many other HD mice with the human HD gene expression profile, but the only one that was overlapping was the one obtained from their mice. So the conclusion was that yes, the BDNF is extremely important for these striatal neurons, and maybe a reduction in BDNF may really be, I don't want to say, of course, it is not the cause of HD, but it could really put the striatal neurons on danger. To the point that several groups in the world are now thinking at the BDNF therapy. And this is maybe where also some of you, I mean, could come to help if you are interested in, because of course now the problem is delivery, how you deliver. I mean, every time, I mean, the problem is always this one, and it is a very important problem. And there are some hopes with this BDNF mimetics. So these are molecules that bind the BDNF receptor and that should mimic the BDNF function. But these are studies ongoing. But at the same time, we wanted to know more about the mechanism, because our idea was that if we understand the mechanism through which normal but not mutant huntingtin affect BDNF production, given that the BDNF is so important, maybe we could come up with some other ideas. And I told you that huntingtin regulates the BDNF production by increasing BDNF gene transcription. And I will make a long story short, but basically we discovered that wild-type huntingtin increases the transcription of the BDNF gene by speaking preferentially to one element of its promoter. The BDNF gene is rather complex, as all very important neuronal genes, and from here to back is the regulatory region, and in this large regulatory region you see that there are 8 elements, and these are exons that are transcribed but they are not translated. Okay, the protein is only made by this 9th exon, and instead these are regulatory elements, as I said. I mean, these are transcribed in messenger RNA, but then they are spliced to the 9th one, and the protein is made always and only by this exon. But Toni Timmusk in Finland has discovered that upstream of each of these elements there are regulatory sequences that work independently. This happens because, of course, you need a very precise way, I mean, to regulate the transcription of this gene depending on the developmental time, cell type, and so on and so forth. And we discovered that normal huntingtin actually speaks preferentially to an element that is located in the promoter region of exon 2, and when normal huntingtin is there, exon 2 is active, therefore there is splicing to the 9th exon, and the BDNF gene is transcribed, the protein is made, and this is the mechanism. And to tell you and to show you just one data that indicates that indeed that this is the case, basically these are again the mice, and now I will show you one data that deals with some of these exons. Given that they are transcribed, of course we can measure messenger RNA transcribed, I mean, from these exons, either in cells in vitro but also in mice, and just look at some of them. And as you can see here, these are the normal mice. These are mice that overexpress wild-type huntingtin, and these are mice that overexpress mutant huntingtin. These are some of the exons that I'm going to show to you, and just focus on exon 2, okay, and focus on the cortex. Exon 2, and this is exon 1, 2, 3, 4, okay. Exon 2 is barely transcribed in normal animals. There is more exon 2 made in mice and this is cortex. I mean, can you imagine? So, okay, there is in the cortex, in the presence of overexpression of wild-type huntingtin, there is more exon 2 produced, and this exon 2 is absolutely gone in mutant huntingtin mice. Okay, and this is just to show you, I mean, these bands. We did another experiment, because we wanted to link, I mean, this huntingtin function as a trigger of transcription of exon 2, and I will come to the second on the reason. But just, I mean, is this link real or not? This is another experiment, and this is done again in cortex, but these are mice that are conditional knockout. The ones before were overexpressing wild type or mutant. These are mice that are depleted of normal huntingtin in cortex. And again, I mean, we measure the BDNF. This is total BDNF messenger RNA produced from exon 9, okay? So this accounts for all the regulatory elements. And, okay, the result was good because, as you see, I mean, as you go from 2 copies to 1 to 0 of wild-type huntingtin, there is the usual progressive decrease in BDNF level. But more interestingly, if we now measure how much messenger RNA2 that we believe, was the target, or messenger RNA 3 or 4 is produced in these mice, let's look at the data. These are the normal mice, and these are individual mice. Mouse number 1, oops, come faccio a tornare indietro precedente? Okay. Mouse number 1 or number 2, and these are 3 mice for the heterozygous knockout, or 3 mice for the homozygous knockout. And this is how much messenger RNA2 is produced in cortex from normal mice. This is messenger RNA2 in cortex from 3 heterozygous knockout and 3 homozygous knockout. And it is clear that if you remove normal huntingtin, there is a reduction in the level of messenger RNA2. So yes, normal huntingtin stimulates the transcription of the BDNF gene by speaking preferentially to exon 2. How exactly? No, because if we understand how, we may think at strategy, to recover that dysfunction. Again, we discover that upstream of this exon 2, there is a silencer element. And since huntingtin is increasing the transcription of the BDNF gene, it means that huntingtin should silence the silencer. And indeed, we demonstrated that this was the case. And to make a long story short, this is how it works. Normal huntingtin sequesters in the cytoplasm a critical repressor factor that would otherwise enter the nucleus and would sit at the level of this silencer element, activating the silencer and therefore shutting off BDNF gene transcription. But when huntingtin is there, rest is sequestered, it does not enter. The silencer is inactive because there is no complex, no repressor complex to activate the silencer, and the BDNF gene is on. When mutant huntingtin is there, rest enters pathologically into the nucleus. The silencer, the repressor complex builds up at the silencer element, and the BDNF gene transcription is off. Therefore, normal but not mutant huntingtin silences the silencer in the BDNF gene, and this is how it works. And a step forward was the discovery that the BDNF is not the only gene carrying a silencer in its promoter, but there are more than 1,000 very important neuronal genes that carry an NRSC, this is an acronym for the silencer, NRSC element in their promoter. Therefore, the question is it possible that normal huntingtin, in addition to controlling the transcription of this BDNF protein, which is so important for the striatal neurons, is also controlling the transcription of so many other NRSC-controlled neuronal genes? Of course, we didn't check all of them, but many of them, indeed, are affected in the presence of normal huntingtin or mutant huntingtin, to the point that we now believe that there is in addition to the BDNF loss that could be cured, possibly with BDNF delivery, there is a generalized pathology, I would say increased silencing of this NRSC regulated gene in HD. How can we rescue this NRSC pathology? And this again, these are unpublished data, and I'm showing this data to you for the first time, and maybe you have inputs, I hope, to give. Because, again, when we have to cure or provide something, then there is always the issue of delivery. So how would you rescue this NRSC pathology? At the moment, we're playing with cells in vitro, and we developed what we called the REST toolkit. So we need to find ways to interfere with this pathological entry of REST into the nucleus of HD cells. How can you work? You can work with the REST dominant negative constructs, or you can work with REST HRNA, so RNA interference, or with ODN decoys. And we did some work in vitro. I will go very briefly through it. And of course, the most difficult part is to try to deliver this in vivo and to see whether we can rescue this NRSC pathology, which means, loss of very important neuronal gene. I think I will show you just one. So, for example, if you express REST dominant negative in cells, we are in vitro here, and these are cells, these are the mutant cells. You can see, if you look at the BDNF and you express the dominant negative REST, you can see that you you increase very much the BDNF level in HD cells. And without going into the detail, if you look at the chromatin immunoprecipitation and you look at your NRSC sites in the presence of the dominant negative rest, basically you see that the NRSC of the BDNF is now free from the pathological occupancy of rest, because you see that the colon is low. And the same for all the other elements, to the point that we also measure the BDNF protein, not only the messenger RNA, and we can see that with dominant negative rest, RNA interference, and decoys, we can rescue this phenotype. And of course, now the critical thing is to do work in vivo. If I have 5 more minutes, 2. Another possible target in Huntington disease is, so this is the reason why I mentioned also the cholesterol, because there is a link between BDNF and cholesterol, and this link was published in 2007, but many years before, Marta Valenza in the lab started feeling that there was something wrong in HD in terms of cholesterol. And everything started from microarray study, because here we basically used again these HD cells, so these are in vitro models, and we found that every time that mutant huntingtin is there, not only there is this BDNF phenotype and all this NRSC pathology, but there is also a loss in the cholesterol level, and the entire pathway, biosynthetic pathway, cholesterol biosynthetic pathway, basically is shut off. And these are the data that we have accumulated over the years. And this is a pathway that leads to the cholesterol. And of course, I mean, we are dealing with the mechanism also here, but I will skip this just to show you the in vivo data. So is it true that there is a cholesterol pathology in the brain, in the HD brain? And just to remind you very briefly, of course, I mean, when we think at the brain cholesterol, I mean, this has nothing to do with the peripheral cholesterol, okay? So these are 2 separate compartments. And the cholesterol in brain is synthesized locally, okay? It is a different story. And of course, I mean, if there is a pathology at the level of cholesterol level in the HD brain, I mean, you can imagine, I mean, you have this mutant protein, you have the loss of the BDNF, and these neurons may suffer also because their membrane or synaptic activity is affected due to reduced level of cholesterol. And indeed, these are 4 different HD mouse models, HD models. These are animals. This is one animal, and as you can see, in these R62 mice, there is a reduction. In this case, we're measuring the precursor of latosterol level, okay? But it is the same for the cholesterol. These are mice that overexpress mutant huntingtin with 46, 72, and 128 CAG, and you see that there is a progressive reduction in latosterol. The same is true in the HD rat model, and the same is true in these HD knock-in mice. So again, I mean, when you test your pathway or target in so many mouse models and you find consistent data, I think this is something to follow with great attention. So what we want to do here is, and this is, I think, my last slide, there is a lot of work ongoing in the lab, and in 2005, we actually demonstrated, this was one of the first data, we demonstrated that if we transfect striatal neurons with mutant huntingtin, and of course we cause a pathology in these cells, but at the same time we provide the cells with cholesterol, you see that there is a gradual increase in survival. Okay, this was, one data, it was put there in the paper, but of course now we want to follow more this, and I'm very glad that this collaboration with Barbara Ruozzi and Giovanni Tosi here from Modena, because now with them we have the possibility to deliver in vitro, and we hope also, the plan is also in vivo, cholesterol through different ways, through liposomes and many other possibilities. And this is a very preliminary data that we just got this week. And these are liposomes, this is one time point. And of course there is a lot that has to be done, but this 1 micromolar cholesterol given to cells that overexpress mutant huntingtin, I mean, really makes something very important to these cells that we know very well that they are suffering a lot, but when cholesterol is given through liposomes, something is happening. So of course we will follow this very precisely. And of course the next step is also dealing with nanoparticles. So we're very excited about this collaboration with this group, and this is my group, and the several collaboration. I didn't have the time to mention all of them, but of course all this work is done by these people in the lab, and also thanks to several funding agencies. And after so many slides dealing with the cells and mice, I want to present to you my favorite species, which is Dictyostelium discoideum, which is the first multicellular organism that appeared on Earth 250 million years ago, and we believe that Huntington was born there with this species. Thanks for your attention.
Thank you. We thank you very much, Professor Cattaneo, for the very interesting lecture. And the section is open for questions. Yeah.
Thank you very much. During your talk, my mind kept asking the question, is there a critical period? Is there a window in development where all this is happening, after which it's not a problem and before which it's not a problem? Do you know anything about that in any of the models?
I would say that, well, whatever we look at in mice, okay, at least at the molecular level, we find defects early on, even in embryonic stem cells. Okay, so this can tell you, would tell you that, yeah, there is probably cell-autonomous immediate dysfunction caused by mutant Huntington. The question is how it is that patients, human beings, I mean, can cope for so long with this mutation, and then this disease appears when they are 30 or 35. And you, I mean, mentioned the word development, and this is indeed very interesting because I would say with some caution that there is now the idea, or there is now the idea that maybe Huntington might also be a developmental defect, developmental disease. No, and there are some data that have been presented recently from University of Iowa that had the opportunity to do MRI scan of kids that carry the HD gene. So they managed to do this. Of course, I mean, there are a lot of ethical complications to that, but they managed to do that. These kids were 30 years away from the disease. So they were, well, okay, they found that there are already problems, at least at the level of MRI scan. So I think we should reconsider. But then even if there are developmental defects, yet, okay, they can cope, I mean, for a number of years. But this tells you that the window of intervention maybe is larger than we thought.
Thank you for this very nice story. Have you considered injecting in mice LDL or high levels of LDL, where you would have cholesterol plus apolipoprotein? This is transported into brain via the blood-brain barrier.
We are thinking at that.
Of course, you have the risk of atherosclerosis, but...
Yeah, exactly. So indeed, I mean, touching the cholesterol is a bit tricky now, and we are always worried that, yeah, you compensate this and you cause other problems. So this indeed, I mean, crosses the blood-brain barrier. I mean, this, and I think, yeah, we are considering this, but for the moment, probably we are trying to go, I mean, with intra-brain delivery. No, but let's say this is something that, you know, I'm not answering because I don't have the full answer for you. Sorry.
My question has to do with the strategy that you you have suggested for, let's say, for the therapy, okay, which is to target cholesterol in the future, of course, to target cholesterol in a very specific part of the brain, because I couldn't realize, because you showed so many slides that I couldn't follow everything, but in what I understood is that cholesterol is missing at least or in cortical neurons or in striatal neurons, or both.
I would say almost everywhere. all brain regions that we tested.
Yes, but anyway, the strategy with cholesterol is to reach those, at least those areas. So if you have the tool to reach those areas, why don't you target the areas directly with BDNF?
Okay, yes. Indeed, I mean, we're not doing the BDNF delivery ourselves. Other people are testing this, but there are problems with BDNF delivery. The protein is sticky, doesn't distribute into the tissue. If you use viruses, in the viral vectors, I mean, to deliver the BDNF, of course, there are problems associated to a viral vector. People have done adeno-associated viral-based BDNF delivery, and it was found that this is very toxic. The BDNF was delivered to a very high level, but it was also retrogradely transported to the nigra, and this caused toxicity. So what I learned that BDNF, I think it is a fantastic protein. Now it is one of those molecules that, of course, our neurons depend on, but maybe because of that, I mean, you really need to define the dose, and defining the dosage and the distribution is indeed a critical problem. So I think at the moment, to my knowledge, there are no clean strategy to deliver the BDNF, to the point that people are thinking more to the BDNF mimetics, the TrkB agonist. But that is, of course, that would be wonderful, I mean, to deliver the BDNF.
Okay.
Thanks.
I have just one general question. But how easy is it to do a correct diagnosis of this disease in comparison with other neurodegenerative diseases?
It is very easy nowadays because, of course, you do a PCR and you just look for your CAG number. And, of course, the patients know that in addition to receiving the information on whether or not they have the disease, they have the mutant gene, they can also receive the information of how many repeats do I have. And of course, I mean, this is a very difficult information to give to the patient because they also know that depending on the CAG size, they can also more or less, so it is not strict, but they can also more or less guess about onset. So it is...
Okay. And actually, which kind of therapy we have for this?
Oh, antidepressants. Yeah.
Nothing.
Neuroleptics, nothing. Yeah. Diet, because it increases BDNF, but they are already very thin. Or exercise, but of course, I mean, they do a lot of movements. So what do you do? No, they are unfortunately abandoned. I mean, from
some kind of gene therapy by not viral vector, some kind of other vector to have the expression in BDNF. Polymeric, for example, or liposomes, or other gene therapy.
This is something that with Giovanni and Barbara we are thinking at, and there are. So my belief is that there are some interesting pathways that can be hit, I mean, therapeutically, but there is a problem of how you provide this therapy. So it is still, I mean, something that has to be worked out.
Okay, thank you.
Thanks a lot.
Thank you again, Professor.